All right. Well, my name is Nana Torpe, and I'm a PhD student at the POCA group at the Biotech Research and Innovation Center. So I'd like to talk to you about Elegans and the approach and technology that we use daily in our lab. So first, just what is an model organism? So we use it as a species to study and understand biological phenomena, and also we have the expectation that discoveries made in the model can be translated into other species. So what are the important characteristics of a model organism? So in Elegans, we prioritize that it has a short life cycle. It can be easily genetically modified. The size really matters, especially if you're working in a lab. You can't have the size of an elephant moving around. Also, the mechanisms needs to be conserved to use it to translate into a higher species. Also, it just needs to be cheap and simple. And also, we don't have any ethical problems, as they were discussing before. We don't have to apply for any legislations or get approval from any of the government. We can just do whatever we want with the worms. And these were the criteria that Sidney Brenner thought of in 1963 when he was looking for a model that contained all these criteria. So he was looking for a small and simple organism which had the complexity to model other organisms and also model the higher organisms. So even though C. elegans is quite simple, it actually has a very developed nervous system and a digestive tract, and also a germline as well. So he wanted to use it to have mutations and then look at the phenotype that the mutation caused, and then to find and dissect the function of the gene. And just briefly about C. elegans, so it's a free-living nematode that lives in soil and feeds on microorganisms. In the lab, it's an E. coli, and we can grow it on petri dishes, where we can have up to 10,000 worms in one plate. So the N numbers for statistics are really high very easily. It's very small, it's 1 millimeter long, so it's the size of a grasshopper brain, as we heard previously. So it's easy to handle, and also it's transparent all the way through embryogenesis and adulthood as well. So it's easy to visualize in a microscope. The lifespan is very short, 2 to 3 weeks, and also the generation time from its hatch to its adult is only 2 to 4 days. So which means that the cycle of an experiment can be increased very rapidly. So the genome size is quite small, so it's only 1/30 of the human genome, and also it's the first multicellular organism to have its genome sequenced, which also means that that is one of the most annotated animals so far. So briefly around the C. elegans, the life cycle. So as you can see here, when the adult worm starts laying eggs, within 9 hours it will have gone through development of the embryos, where it will be hatched as an L4. And here, depending on the environment, if it's a hostile environment, so if it's too crowded, if there's starvation, if there's high temperature, the worm can go into a dour state. That means that it closes off all cavities, and then it can actually survive for up to 4 months before it started dying. And once the environment changes, if it becomes food or if it becomes more favorable, the worm can go into, then continue into the L4 larval stage, and then become an adult, and then start all over again. If here at L1 the environment is nice and there's food enough, it just continues to L3, L4 up here, and then it starts again. So it's only 3 to 4 days. So the experiments can be very rapidly performed. So briefly, that's the hermaphrodite and a male, and also crossing, and a larval and an egg. So you have an idea of what they look like. So this is in the laboratory on E. coli. So the advantages of the worms are quite many, we think, at least in our lab. And the first one is that they can be handled like a microbe. So they can live on agar plates, as I described, on E. coli. And they can also be kept in liquid medium. And they can be frozen as stocks, which is a really great advantage. Because every time you make a transgenic strain or a mutant, then you can freeze it down. And then you can thaw it later if you need it on a later time point. Also, we have a CDC knockout consortium that makes knockout animals that you can order them and then freeze them down as well. And again, there's no ethical problem, so you can do whatever you want. Also, it's very amenable to genetic analysis, so it can self-fertilize, which makes it easy to isolate recessive mutants. And also it crosses with males, so you can make a knockout animal within 3 weeks, which is very fast. And you can also easily make double or triple knockouts of genes of interest. Also, the genome sequence is known, which means that we can have genome-wide sequencing, which I will turn to later. Also, the small size, as you can see here, means that we can, and the transparent body means that we can easily image, so with different GFP markers, which are driven by, so genes driven by GFPs for specific cells or in tissues. So it's very easy to visualize in a microscope. Also, it has an invariant cell number and lineage, which I will come back to later, why that's a great advantage. And also, it's very easy to make mutants through mutagenesis, DNA microinjection, or RNA interference. So the invariant cell lineage, it looks really weird. Oh, okay, Mac, I guess, was mapped by Joel Sutton when he was a postdoc. So basically, he was sitting in a dark room for 1.5 years, drawing out in a DIC where the different cells moved. So it's quite immaculate. And as he says, a simple analogy is to imagine that you're watching a bowl with hundreds of grapes, trying to keep your eye on one grape as it and others move around. So, and this is what when he finally mapped everything, so we have the entire lineage and cells, which is very important. When you can have a GFP marker for example, muscle, and then you can look and screen for mutants of cell division in the muscle lineage, for example. Yeah, and again, back to the no ethical problems. So here, what we can actually do to create mutants through mutagenesis is that we can take a whole pile of worms here and then treat them with ethyl methane sulfate, which is a mutagen, and you can produce random mutations. Then you let them self-fertilize, and then you have mutations which are inherited through the germline. And here you can then pick all these worms, different plates, and then screen through for a phenotype of interest that you have down here. And for this we use 4 genetic screens, which is basically used to identify genes or a set of genes responsible for a particular phenotype in an organism. And that's mostly used in developmental neurobiology, where you have a GFP marker for example, dopaminergic neurons, where you then screen for lack of expression in the neurons. So you can screen through a lot of worms, as I'll explain later. And also you can have a suppressor screen where you identify other interacting components in a genetic pathway by identifying second site mutations that suppress the mutant phenotype. And that's mostly if you have identified a phenotype like axon guidance, where you have a defect and then you want to see in which pathway that is working. So you look for a suppressor of that phenotype. So you basically treat your mutant with EMS and then find the worm that has a phenotype where it's suppressed. And then you can do whole genome sequencing, where you just send it off to a sequencing facility, and then that will give you the target identification, since you can compare the entire genome of your mutant with the wild type animal. And before this was done manually, and I've done it myself, and it takes really a long time. But now we got this. It's very exciting. It's a worm sorter, so it's kind of like a giant fax. So you can just pull through the whole worm, and you can screen 80,000 mutagenized genomes in 25 days, which gives you around 18 to 20 mutants. And what you basically do is you treat them with EMS, and most of the time it contains a GFP macro of the cells you're interested in. And then you just run it through, and if you look at mutants where the gene is silenced, then the worm machine will pick that out, and then you can just continue working with that. And most of the times we go for whole genome sequencing after that. So it really gives a great advantage to dissect the genetic pathways. Okay, again look weird. So what we do afterwards is to further investigate the mutants that we have, and we can do that by mutant rescue and transgenics, which is done with injecting DNA into the cytoplasm of the gonads, as you see down here. And that we pass the germline in the form of an extra chromosomal array. So you can use that for rescue of a mutant phenotype using a wild type copy of the gene, or have a gene expression pattern analysis with a promoter fused to a GFP. And also you can use it to interfere with the biological process by expression of wild type or mutated genes. So just an example here, there's 3 approaches. So one here is the reported gene fusion. And there's also in situ hybridization using mRNA or immunofluorescence with specific antibodies. It's quite easy to stain as well, C. elegans. And here's an example from my own work where I have the promoter driving my gene of interest, and then driving GFP. So as you can see here, it really gives you quite a good picture of where your gene is expressed. Also, C. elegans can be used to study cell division, and you can see here a fluorescent micrograph of a wild-type C. elegans embryo of the metaphase of the first division. And since the embryo is transparent, you can really see here how you can look at an entire organism, how it's developing through time and all the way up to when it's hatching. So just briefly, a summary. So we think that C. elegans is a very powerful organism, especially because it's powerful genetics, and it's enabled dissection of disease mechanism in several fields, and also the fact that 75% of human disease genes have a potential C. elegans homolog is very powerful. And as I showed you, it's very easy to make mutants. And also the strength of forward genetic and suppressor screens makes it very easy to identify novel players of genetic pathways or genes responsible for a particular phenotype. And that, combined with the whole genome sequencing, give us a really good tool for finding new players in different disease pathways. And I would like to thank you, and also the tiny worm for putting up with us every day. Yeah, thank you.